Hi everyone, I'm Hannah and I'm going to talk to you about what I did this summer. It's estimated that 1 in 2 Canadians will develop cancer in their lifetime. Of those diagnosed, a fifth of them will be men who develop prostate cancer and a fifth will be women who have died from ovarian cancer. As many of us are aware, it is a big issue in the world and is ever-changing as we study it. The newer field of nanomedicine has brought to the forefront how nanoparticles have unique properties and abilities which may be beneficial for targeting cancer cells. So, my research investigates how toxic these particles are to human cells. I'm using prostate and ovarian cancer cell models, as well as a human kidney cell model, to examine how different coatings on the outside of gold nanoparticles affect how they interact with cells. I also want to see if the different cell lines react differently or the same to each coating. The reason why the coating is so important is because the first contact the particle has with the cell is through the coating. And if the body doesn't like it, then it's going to try to get rid of it. The gold nanoparticles I use are 5 nanometers, which to put in perspective is about 20 times smaller than a strand of hair. The particles are coated with PEG, a very biocompatible molecule, which should help them be uptaken by the cells. I use 2 different kinds of PEG. One is terminated with a carboxy group, and the other is terminated with a methyl group. I also use gold nanoparticles coated with DMAP, which is a very toxic compound. How I do all this is outlined on my slide. So first, I start by maintaining the cells and making sure they're happy and healthy, and let them grow in a petri dish or flask until they're about 80% of the bottom of the plate. Then I dislodge them, or passage them, as my slide says, and I replate them into a 96 well microplate. I let them grow for 48 hours, which lets them again expand and grow into their new surroundings in the well and fill 80% of the well. Then I treat them for 24 hours with different gold particle coatings at different concentrations, and after the 24 hours I remove the treatment and run a multitude of different assays which let me assess different parameters of cell health and how cytotoxic the particles are to the cells. Through this, I've noticed and gathered that each particle exhibits a different profile for toxic effects on each cell line. I've also noticed that across cell lines, the particles are acting differently, which suggests different routes for cell death dependent on the particle coating and cell line.